After paramyxovirus, we have orthomyxovirus, and the typical example of orthomyxovirus is influenza virus. So we have known that orthomyxovirus is a negative single-stranded RNA having enveloped and non-infectious. So influenza, an example of orthomyxovirus is influenza, very common, and the viral genetics is very much asked in the exams. So influenza has the envelope, and in the envelope of the influenza we have the hemagglutinin and neuroaminidase enzyme. So hemagglutinin is binds to sialic acid and promote the viral entry into the host cell, while the neuraminidase enzyme, it helps the virus, it helps the release of progeny of the virus after replication. So the treatment of influenza virus is relatively based on these two, entrant and exit inhibitors. So medication which are the neuroaminidase inhibitor act on the neuroaminidase and inhibit the release of the progeny, and includes oseltamivir and zanamivir. And we also give sportive treatment. Then we give flu shots every season whenever the outburst of a new strain happen. So we use a killed vaccine. Most frequently we include the strain of that particular virus in the respective season, and kill this and make it inactivated so that it can generate immune response in the body. So in the light attenuated version, it is given in the intranasally, and it is temperature sensitive, means it is activated only in the nasal part and inactivated or destroyed in the lungs. So if a patient is contracted influenza virus, then it has the increased risk of susceptibility to the superinfection, increased risk of susceptible to superinfections from Staph aureus, strep pneumo, and Haemophilus influenza. So this is all the discussion. The important educational objective from the U world is that the major important determinant or the factor which the major adaptive immune response of the body which prevent reinfection is the generation of the autoantibody against the hemagglutinin part of the influenza virus. It helps the body to generate a major adaptive immune response and prevent reinfection. So according to the viral genetics of the segmented viruses, in the segmented viruses we have the B, O, A, R, Rio, and then Orthomyxo, Bunia, and Arena. They are the segmental viruses. If a cell is co-infectedly with two different strains of virus, like a virus, for example influenza virus or the rotavirus, the patient or the hantavirus in the Bunia section, hantavirus, if the patient is infected with two different strains of these segmented viruses, like a wild one and somewhat a human strain or avian strain, then in the host cell the two viral RNAs, segmented viral RNAs, the RNA segment of these viruses undergoes genetic reassortment, and this reassortment goes to the next progeny, and the progeny is very much a novel virus which can cause a new epidemic and pandemic. So it is the major cause of the global pandemic and epidemic, and this type of viral genetics is known as antigenic or genetic shift, and shift is very severe like the coronavirus. So this shift is happen when the two different strains of the segmental viruses undergo genetic reassortment. Next, if the hemagglutinin or neuraminidase enzyme genes randomly mutated in a host cell which have developed adaptive immune response to prevent reinfection from a same strain, then the this random mutation can cause alteration in the entry and exit protein, surface protein, and it causes local or epidemic infection in the particular disease, like it happens annually during the seasons of winter season. It happens every year, and we have to give the new flu shot strains, new seasonal flu shots, in order to cope up with that infection. So this random mutation increase the infectivity of the virus, infectivity, and it also helps to evade immune response of host. So this is the very much typical mechanism of orthomyxoviruses, segmental viruses, how they reinfect again and again in every season.